Hello, this is Soha Hassoun from Tufts University. I'm here to tell you about our work entitled Pathway Activity Likelihood Analysis and Metabolite Annotation for Untargeted Metabolomics using Probabilistic Modeling. This work is in collaboration with Professor Liping Liu and two of our graduate students, Ramtin Hosseini and Nida Hassanpour. A unique characteristic of metabolomics is its ability to reveal cellular functionality. We are already seeing some remarkable works of how metabolomics is allowing us to take a panel of biomarkers and making them clinically relevant or applicable for studying cellular processes. A recent example is a study published in Autism Research, where scientists at NeuroPoint DX, in collaboration with others, reported new findings from the Children's Autism Metabolome Project. The researchers identified unique metabolic signatures in over 50% of the children with autism in the study. This is an important step towards the goal of developing a panel of metabolomics tests that could form the basis for a biological screen to analyze the risk of autistic spectrum disorders. There are many other studies, including those focused on the microbiome and on exercise, that have shown metabolomics effective in identifying biomarkers and linking them to health and disease. Advancing metabolomics research to the point of broad use in healthcare and in other fields, such as biotechnology, will require significant improvements to the interpretation of metabolomics data. My presentation is about the interpretation of untargeted metabolomics data in the context of the biological sample. Given a set of metabolomics measurements and a network model corresponding to the biological sample, I'm interested in investigating two questions. Which metabolites are present in the sample, and which pathways are active? Here, we call a pathway active if the likelihood that the pathway generated the observed measurements is above a particular user-defined threshold. Our goal in this work is to answer these two questions using probabilistic modeling. Unlike prior techniques that focus on differential analysis between a perturbed and a reference sample, here we focus on analyzing a single sample. In this work, we capitalize on our understanding of the uncertainty in mapping measurements to metabolites and metabolites to pathways. Here we have a snippet of a network with two pathways, pathway 1 and pathway 2. Each pathway is associated with several metabolites. Metabolites are represented as circles, while some metabolites, such as J1, is associated with a single pathway. Others, such as J2 and J3, are associated with more than one pathway. When performing metabolomics, we observe several masses. In this picture, masses are represented as squares. Some masses, such as W1, can only be attributed to one metabolite. Other masses, such as W2, can correspond to multiple metabolites. We can all agree that we have more certainty in attributing W1 to the activity of pathway 2, more so than attributing W2 to the activities of pathway 1 or pathway 2 or both. So the idea of this work is simple. We would like to use probability to capture the uncertainties in mapping measurements to metabolite and metabolites to pathways. To solve this problem, we will use probabilistic modeling. Probabilistic modeling involves several steps. First, we define a model using random variables. Our model captures relationships between pathways, metabolites, and their masses. We then make assumptions about distributions on the random variables. Once we have a model and model parameters, we can then form hypotheses and calculate the probabilities of the hypothesis given the observed data. We could use Bayes' rule to compute such probabilities. We implemented our probabilistic framework using PyMC3, a Python package for Bayesian statistical modeling. We call our tool puma. We analyzed 3 datasets. I will highlight some results. We evaluated metabolite annotations obtained using puma for LCMS datasets collected by a collaborator for the Chinese hamster ovary cell. Our collaborator's lab analyzed the CHO datasets thoroughly. After pre-processing the metabolomics datasets, they provided Metlin and HMD matches for each measurements when possible. They also use their tool called biocan, which aggregates measurements from spectral databases, different tools such as MatFrag, and network connectivity. Biocan predictions were experimentally validated for 50 predicted annotations against chemical standards. A subset of 37 out of 50 compounds were confirmed present based on the standards. Thus, biocan's precision on this dataset was 0.74. Puma's precision for this set is 0.833. Puma achieves a significant additional improvement of 0.093 over biocan. Puma thus has high precision, returning substantially more relevant results than irrelevant ones. This work presents a probabilistic paradigm for metabolomics analysis. We can determine pathway activity and annotations from all the measurements and the network topology, all based on inference. We provide significant improvements over an aggregator tool such as biocan. For more details, please read the detailed paper, which was published in Metabolites in 2020. The code is available on GitHub. Please give it a try. I'd like to thank the students in my lab and my collaborators, including Professor Lipping Liu, who was instrumental in developing this work. Thank you.